Well, believe it or not, here we are, the last recorded lecture for this entire semester. It's been a pretty long road, but I think it went well. Obviously, we'll meet quite a bit more in class, but this is the last official lecture, so this is the last lecture of Unit 3, and the end of this lecture marks the end of the material, both for exam number 3, as well as the end of the material you're responsible for on the cumulative final. This is the second part of Chapter 25, 14 pages from that chapter, pages 701 to 715. Today we'll first talk about the effects of non-random mating on a population and how this pertains to Hardy-Weinberg equilibrium, and then other things as well that change allele frequencies, such as mutation rates, migration of a population, population size, etc. So we talked in the last lecture about the Hardy-Weinberg equation, the Hardy-Weinberg law, and Hardy-Weinberg really takes care of everything from a population genetics perspective. It takes the laws of Mendel's work, the laws of dominance and recessivity, 2 alleles per individual, and combines those laws mathematically with the genetic behavior of a population. Really what the Hardy-Weinberg law does through its equation is it ties allelic frequencies with genotypic frequencies, and this allows for prediction. As long as the population is in Hardy-Weinberg equilibrium, we can calculate and predict the number of homozygous individuals and heterozygous individuals genotypically, knowing nothing more than the frequencies of the alleles in the population. And we did all that in the last lecture. However, and we can't say it enough, in order to do this and use the Hardy-Weinberg equation with high fidelity, to use it and trust it, the population being studied must meet 3 distinct criteria. It has to be very, very, very large. Remember, the true requirement is infinitely large, although that's never reachable, but it has to be a very, very large population. Mating among the different individuals of the population must be as close to random as possible, and there must be no mutations, migration, or natural selection of any kind. Again, infinite populations is unachievable, no mutations is unachievable, but we want these criteria to be as close to Hardy-Weinberg assumptions as possible. Very, very large, very, very non-random mating, and as small an amount of mutation, migration, and evolution as possible. What we're going to talk about in today's lecture is why these criteria are so important. We're going to see what the effects are on a population as it pertains to allelic and genotypic frequencies when these rules are broken, when mating is non-random, when there are mutations, migration, when there is natural selection. We're going to focus less on the math today. Really, the only math you need to understand thoroughly is the math of the Hardy-Weinberg equation itself, the math that we covered in the last class. We're going to focus more conceptually on how these broken rules affect Hardy-Weinberg ideas and concepts. So first, non-random mating. There are 2 broad categories of non-random mating in any species and for any trait. Positive assortative mating is when individuals tend to mate with others that are like them. So positive assortative mating means that 2 mating individuals mate non-randomly and choose one another because they are more alike, as we see in this image here. And then negative assortative mating is the opposite, of course. This is when individuals tend to mate non-randomly. They choose partners that are not like themselves. So negative assortative mating, there's still choice, it's still non-random. Partners are still picking each other for mating events, but they're picking each other on their differences, not their similarities. Like population genetics itself, these different categories only pertain to individual traits, phenotypes, or genes, not individuals as a whole. In other words, 2 individuals can exhibit positive assortative mating for one trait, say eye color. 2 blue-eyed people choose to mate with each other because they share the same eye color. That's positive assortative mating. And these 2 individuals also choose to mate with one another non-randomly. They've selected each other due to negative assortative mating for hair color, completely different trait. Same individuals, different trait. So one might be black-haired and the other blonde-haired, and that attracted them to each other. That's negative assortative mating. More broadly, in general, humans tend to show, on average, positive assortative mating for height. So individuals who are tall tend to select mating partners who are also tall. Short individuals tend to prefer to find shorter people attractive. But it goes without saying, gender is a phenotype. We as a human species, by definition, show negative assortative mating for gender. If we're going to mate, we need to mate with somebody of the opposite gender. That's negative assortative mating because opposites are attracting. The example we're going to use for non-random mating is inbreeding. It is the most pronounced and dramatic form of non-random mating. That's why we are going to use it. But more importantly, inbreeding affects all traits, not just a single trait, and so we use it for that reason as well. Inbreeding is defined as positive assortative mating for relatedness. Individuals are choosing one another because they are related. That's the commonality they have, so it is positive assortative mating. But again, unlike all other non-random mating categories, inbreeding affects all genes. If we showed positive assortative mating for eye color, that positive assortative mating would have an effect on how the eye color gene is transmitted into the next generation, but it would only affect the trait that was involved, eye color. Inbreeding is used as our most dramatic example because it affects all genes, because once you're related with someone else, you have more genes in common with them than not on a genomic level, genome-wide. And so this is the only non-random mating that affects all genes instead of affecting only the genes that contributed to the trait being mated for. So positive assortative mating for relatedness or inbreeding skews genotypic frequencies away from the predictable p squared, 2pq, and q squared that we could calculate using Hardy-Weinberg equations in the last lecture. Being a little bit more specific, what happens when inbreeding occurs is that we see an increase in the number of homozygotes in general. This is throughout all genes for the entire genome, and a decrease in the number of heterozygotes. We can see that here. This is the most severe case of inbreeding is self-fertilization. We can do that with plants. And we go from a very nicely distributed allelic frequency of 0.5. If we have allelic frequencies of 0.5 for two alleles, we see 25% of our population is homozygotic dominant, 25% is homozygotic recessive, and the remaining 50% are heterozygotes. After just a single generation of self-fertilization, again, self-mating, the most extreme case of inbreeding we can find, we've halved the number of heterozygotes from a half to a quarter. That's in one generation. And we've bumped up the homozygotes to 3/8 instead of 1/4. We have it again in a third generation. 1/8, 7/16, and 7/16, 1/16, 15/32, and 15/32, until eventually we will approach 0 heterozygotes, and half of our population will be homozygotic dominant, and half of our population will be homozygotic recessive, and they will always be that way. Because with positive associative mating for relatedness, these plants will only mate with other plants like them. And so it will always be a homozygotic recessive by homozygotic recessive cross, and these plants will only mate with other plants just like them. And so it would be dominance crossed with dominance. So that's the most extreme case. Now, outcrossing, or mating with individuals outside of the family, a species' tendency to not mate with related individuals, obviously counteracts inbreeding. For most outcrossing species, and ourselves, the human species, is certainly a member of that, inbreeding has the potential to be quite harmful. We are meant to outcross. We are not meant to inbreed. When we inbreed people and other outcrossing species, we boost the percentage of homozygotes for the reasons I just showed you on that figure from your textbook. Inbreeding then increases or boosts the chances that an individual will be homozygotic recessive, because if we're increasing the number of homozygotes in general, half of those homozygotes will be recessive. If we then remember that most recessive alleles are usually of poorer quality, they're usually mutant, in fact, we mean by boosting the number of homozygotic recessive individuals, we're increasing the probability that those individuals will express a weaker recessive trait. These increased appearances of these deleterious traits in these homozygotic recessive inbred individuals is referred to as inbreeding depression, a reduced fitness on the overall population that's due to directly an increase in the number of homozygotic recessive individuals, but that is due to the inbreeding practices. Here we're looking at the number of homozygotes in a population versus the number of generations of inbreeding. We just saw for self-fertilization it takes somewhere in the neighborhood of about 6 generations, a little bit more, of self-fertilizing inbreeding to max out at the entire population being homozygotes, no more heterozygotes at all. For sibling matings, we get close to 100% after about 14 or 15 generations of inbreeding. That's of siblings. So that's not all that many generations. That sounds like quite a bit, but that's certainly within the scope of evolution. Look at this, even for first cousins. When first cousins are inbreeding with one another, this is probably the most likely scenario that you would find in some families and some tribes and some customs of the human species. After about 10 generations of inbreeding first cousins, we get to about 65% of the population is homozygotic. And so half of that, or about a third of the population, is going to be homozygotic recessive. That's a whole lot of unfit individuals showing deleterious traits genome-wide. And the severity of the inbreeding depression correlates with how intense the inbreeding is, or in other words, how closely related the two mating partners are. The effects of inbreeding have been known empirically for millennia. I mean, these go back to, there are biblical references and biblical laws against inbreeding. And because of these deleterious effects that it has on families and on people and our species as a whole, this is probably the main root cause of the social taboo on such practices. In addition, child mortality increases with inbreeding. If two completely unrelated parents mate with one another, this is a single mating event, the rate of mortality for that child up until about age 12 in Japanese children is about 8.2% or so, quite high, but that's the percent. If second cousins mate, we gain quite a bit. We crack the 10% mark on just second cousins. Second cousins! Quite unrelated, but still we crack that 10% mark. And if first cousins mate, we're looking at about 11.5% mortality versus 8.2% in unrelated, just due to a single mating of first cousins. So there's inbreeding depression, which hits the population over many, many generations of inbreeding. But even a single inbreeding event among two parents has a direct effect on their offspring, increasing the chance of child mortality. Inbreeding negatively affects crop yields. This was noted very early on in the human history of agriculture. If we have a very, very low inbreeding coefficient for corn, meaning we intentionally force one corn plant to pollinate or mate with another unrelated corn plant, we see very robust crop formation, very robust corn. An inbreeding coefficient of 1, meaning the two mating plants are completely related. The only way to do this is with self-fertilization of the corn, but you can do that. We see horrible corn yields. The ears of corn are smaller, the kernel production is much less efficient, and we have a significant detrimental effect on crop yields due to inbreeding. Even in mice, we see the same story. We can have captive lab mice. We can force them to inbreed. And if those captive lab mice are then released into a like wild environment, an environment outside of the lab and outside of the cage that simulates a wild environment, those inbred captive lab mice, once released, have a survival rate that is 56%, nearly half as good as their outcrossed non-inbred counterparts. In addition, when those inbred mice were released into the wild environment, they lost a lot of weight due to the stress of release. They never gained it back. The outbred mice that were outcrossed, they lost weight too. Same stressful insult, same stressful environment in a new and wild area. But once they've recovered from that stress, they gain that weight back again. So there's just an overall increased fitness for outbred organisms that far exceeds inbred. So, I shouldn't make it sound like inbreeding is always detrimental. Inbreeding actually can work. It can work well in artificial selection environments, right? When we're breeding cows or we're breeding livestock, we're breeding other agricultural crops like corn that respond better to inbreeding. Inbreeding can work, but it only works if the helpful, positive, healthy alleles are the ones that are artificially selected for and promoted for. while the deleterious alleles are selected against and weeded out of the population. You can achieve this with a very carefully designed breeding program, or you can even achieve this naturally when natural selection weeds out those unfit inbreds, makes it impossible for them to biologically reproduce and get those unfit alleles into the next generation. But it's arduous on the artificial side, and natural selection and evolution tends not to work so quickly and so well from generation to generation. So outcrossing is clearly the more successful strategy for sexual reproduction overall, and that's why we see that most life on this planet does indeed outcross. So that's our example of how non-random mating affects Hardy-Weinberg equilibrium and Hardy-Weinberg math, and why Hardy-Weinberg has as an equation that mating must be random. So let's move on now to mutations. All evolution relies on genetic variation. That's what evolution is. It sees the differences in the genetics and the phenotypes of individuals, and it works to select some and unselect or deselect others. And this genetic variation really boils down to lots of alleles. And lots of alleles are caused by mutations. Never lose sight of the fact that meiosis and sexual reproduction bring existing alleles together in new combinations, and sometimes those new combinations of alleles have different fitnesses, have different levels of survivability and reproduction. But what really drives evolution long term are new alleles. New alleles that have a chance to outperform the existing alleles in terms of fitness. And only mutations make new alleles. So you can think of mutations, things going wrong, like electronics, your electronic devices breaking down in your house. So mutation is a random event. You never know when it's going to come. You never know what it's going to look like. Chances are it's going to make something a whole lot worse, but there's a slimmest chance it might make something better. But it's random. You can't foresee it. Just like something breaking in your house. But the important point here is the more electronics you have, the higher you make the chance that one of them is going to break, right? If you have only one smartphone, there's a chance that that smartphone might spontaneously and randomly break, but it's a small chance. These things are built quite well. But if you have in your house, in your possession, 20 or 30 different devices, the chance that one of them is going to break goes up quite a bit just because you have so many of them where the opportunity for one to break could be there. Alleles work the same way. The alleles in a population with the highest frequencies, and the easiest way to say that is the alleles in the population that there are the most of, get mutated more simply by virtue of the fact that there are more of them in the population. Mutations are random. They occur randomly. And so it's almost like a raindrop dropping on the ground. The more stuff you have outside, the more chance one of those things is going to get wet with a raindrop. Just because there's more places for the raindrop to land, more electronics to break down in your house. So I want you to imagine a gene with two alleles, a very simple two-allele system. There's a functional wild-type dominant A allele and a non-functional recessive mutated little a allele. I want you to imagine that for this gene, there are 45 copies of that dominant A allele and 5 copies of the recessive allele. This would be an allelic frequency of 0.9 and 0.1 respectively. So P would be 0.9 and Q would be 0.1. Take-home message, there's a lot more of the dominant alleles than the recessive. If there's a mutation event, if a raindrop falls out of the sky, it's much more likely that that mutation event is going to occur on the dominant A allele, simply because there are more of those alleles in the population. There's more of a chance that the person with a greater number of electronics is going to have one of those electronics break down because there's just more of them. And when that dominant A allele mutates, it's going to mutate, most likely, to a non-functional recessive A allele. That's what mutations tend to do, make alleles less functional. So this is going to cause a shift from dominant A to recessive A, and it's going to skew the allelic frequencies. If the allelic frequency for the recessive alleles was 0.1, now it's 0.12 because we just gained a recessive little a allele due to this mutation. If another mutation occurs, we're going to mutate another dominant A allele to a recessive A. Now we've skewed it from 0.12 to 0.14. And then also keep in mind where those new recessive alleles are coming from. They're coming from the pool of dominant A alleles. And so the dominant A allele frequency, P, which in this case was equal to 0.9, is dropping at the same time that the recessive A allele, Q, frequencies are increasing. Excuse me. So when P is very, very large and you have many, many dominant wild-type alleles, more of those alleles will be mutated by random chance simply because there are more of them for the mutations to strike. But there are also things called reverse mutations. We've covered these before in this class. Reverse mutations take the mutated allele, in this case little a, and through a second mutation revert it back to being wild-type, dominant A. Those reverse mutations are going to be extremely rare early on when the number of recessive A alleles is low. But as the number of recessive A alleles climbs, it becomes more and more likely for the raindrops to hit those recessive alleles. And when they do, there's a chance that those recessive alleles will mutate back into dominant alleles. Commiserate with that, as the dominant alleles are mutating, their numbers or frequencies go down, and so the mutation rates on those dominant alleles slow down, because there are fewer and fewer of those alleles to be hit with the raindrops. Eventually, the end result of all this is that the mutation rates of dominant A and recessive A alleles match each other, they stabilize each other, they reach an equilibrium. And we mean a true equilibrium. Not that these mutations stop, but a true equilibrium. An equilibrium is when there's an equal amount or an equal flow of something in one direction and the other. So an equilibrium on a ship taking on water is that for every gallon of seawater that comes into my ship, I bucket a gallon of seawater out. The amount of water in my boat stays constant. But it's at an equilibrium because there's a constant flow of water in and water out, an equal flow of both. Same here. There's an equal and constant flow of mutations in one direction and the other. Every time a dominant A allele mutates to being a non-functional recessive A allele, there is a recessive A allele in the pool that is mutated and becomes, again, a functional dominant A allele. So the number of dominant alleles and recessive alleles stays constant, but there is also a constant flow of mutations going in one direction and a balancing flow of mutations going in the opposite direction. That's the equilibrium. So the net direction of mutations, the overall direction of mutations, is zero. Once this happens and this equilibrium is reached, it's possible to find a new Hardy-Weinberg equilibrium where the allelic frequencies are predictable and stable from generation to generation, and therefore you can predict the genotypic frequencies as well from that math. But mutation rates at any gene are low. Mutations are very, very rare, and they're slow. Mutations don't happen often in a single generation. So once that new equilibrium is established, that means there's no more change in the population. That means there's no more evolution. The effect of typical mutation rates, being low and slow, on Hardy-Weinberg principles is very, very negligible. But evolution is low and slow, and so this works well. Populations from generation to generation remain largely predictable because mutation rates are so negligible they really don't affect Hardy-Weinberg math too much. But evolution as a process is slow, and so species still evolve within the confines of these low, negligible levels of mutations because evolution as its own force is moving at the speed of a glacier. So that then is the story of how mutation rates affect Hardy-Weinberg equations and Hardy-Weinberg math. Take-home story for mutations: Hardy-Weinberg law says allelic frequencies are stable from generation to generation. This allows you to predict genotypic frequencies from generation to generation. That's the essence of Hardy-Weinberg law. Now we just saw that before we reach a new equilibrium, what do mutation rates cause? They cause a loss in the allelic frequency of one allele and the gain of an allelic frequency of another. That means allelic frequencies are not stable from one generation to the next. That means allelic frequencies are not predictable from one generation to the next. So that means that genotypic frequencies cannot be predicted from allelic frequencies, and that breaks Hardy-Weinberg law. That's why Hardy-Weinberg assumption includes little to no mutation rates. Another Hardy-Weinberg assumption is little to no migration rates. So let's talk about the effects that migration has on these laws as well. Migration refers to the influx of individuals, which then means the influx of genes and alleles, from one population to another. We call that migration or gene flow. And remember again that Hardy-Weinberg assumes no migration of any kind. When migration occurs, it actually accomplishes two good things for a population. One, it stops separate populations from getting too different from one another. When two populations of the same species remain isolated for too long, it can often lead to speciation, which is that the two populations become two different species, and they can no longer intermate. Migration stops this from happening because it constantly gets those two populations to reshuffle and remate again. The second thing that migration accomplishes is that it brings new alleles in new allelic frequencies into a previously stable population. This allows new fitnesses to emerge, new genotypes to emerge, and this can help the health of the overall population. The simplest migration, the migration we're going to discuss here, is called unidirectional migration. This is where representatives from one population migrate to a second population, but representatives from the second migration don't migrate to the first. So unidirectional. Individuals are only moving in one direction. And what happened here is we have two populations of dots. One population is primarily made up of blue dots with some red individuals scattered in between. The red individuals here are recessive. And we have another population that is mostly recessive individuals. So mostly red dots with a few dominant blue dotted individuals in there as well. Two completely different allelic frequencies in these two populations, but the same alleles are in play. These are two populations of the same species. And in this scenario, a few representatives of population 1, just these 5 individuals, set off and migrated into population 2 and joined population 2. See the effects that we have on the allelic frequencies. Because the allelic frequencies of the dominant and recessive alleles were so markedly different in these two populations, even this small, limited migration event dramatically shifted the allelic frequencies for the dominant and recessive alleles in population 2. In this example, if you do the math, this single migration event has changed the allelic frequency of the dominant allele P from 0.125, or 1/8, to 0.2, or 1/5. That's a huge difference on a single migration event. Likewise, of course, because we always have to equal 1, the allelic frequency in population 2 of the recessive allele went from 87.5% to 80%, a marked drop, because these two populations were so different in their frequencies. So we see a single migration event of just 5 individuals having a dramatic effect on allelic frequencies. If this migration rate continues in this way, in subsequent generations, the populations will what we call homogenize, where the population 2 allelic frequencies will become overwhelmed and overtaken with those allelic frequencies we see in population 1, and population 2's allelic frequencies will then assume the very same allelic frequencies from population 1. The two populations will be indistinguishable from each other. These types of migration events tend to benefit both populations, because at least for population 2, we see an introduction of new allelic frequencies, additional alleles into the gene pool, giving those individuals a little bit more marbles in the bag to choose from, as well as giving more alleles to the gene pool that can be hit with mutations and allow for evolution to occur. Keep in mind, we made the point that recessive alleles are usually deleterious. This was a very, very unhealthy population, if that's the case. Most of the alleles in this population were recessive. So this is an unhealthy population. This shot in the arm, this inoculation of some dominant individuals from population 1 has helped to balance that and get more dominant alleles in this population, improving the health of population 2 as we went along. Also, looking in reverse, we can now appreciate why there can't be migration in populations that conform to Hardy-Weinberg principles because, again, Hardy-Weinberg says that in order to use this predictive math, allelic frequencies from generation to generation must be stable. And if we have these migration events happening from generation to generation, these allelic frequencies are anything but stable. We can't, in other words, predict the frequency of dominant and recessive alleles when individuals are migrating with each generation from one population to the other. The reason we can't do the prediction is because, like any migration event, we never know how many individuals are going to migrate. One generation it might be 5, another generation it might be 8, another generation it might be 12, and each time there's a migration event, the allelic frequencies in population 2 shift. And Hardy-Weinberg says the allelic frequencies are stable so that they can be predictable. Hardy-Weinberg populations are predictable. But until these two populations either homogenize or the migration events stop, the allelic frequencies in this population are anything but predictable. And so they break Hardy-Weinberg laws. Some more factors that affect allelic frequencies and therefore affect the power of Hardy-Weinberg predictions. Genetic drift. Hardy-Weinberg's assumption includes that the population must be infinitely large, or if not realistically possible, as large as conceivably possible. Why? Why is that important? Only for infinitely large populations can you be guaranteed that each and every single allele existing in the gene pool will be passed on by reproduction to the next generation. Mathematically, that's what you need. An infinitely large population assures you that every single allele in the population will make it to the next generation. That's important statistically. Just as only for an infinite number of coin flips, of penny flips, will you be absolutely guaranteed to have exactly 50% heads and 50% tails. Only with an infinitely sized population can you be guaranteed that every allele will make it to the next generation. Once your population is smaller than infinite, you have the potential, for chance and randomness to skew the alleles that are involved in mating events, to skew the alleles that make it to the next generation, and that invalidates the predictive quality of Hardy-Weinberg math. So you flip a coin 10,000 times. Do you come close to 50-50? Yes. Do you come damn close to 50-50? Yes, you do. But is it possible that you had 4,997 heads and 5,003 tails? Quite possible. Is that exactly 50-50, right on the nose? No, it's not. So if you have 10,000 individuals, is it likely that 99.5% of the alleles in that gene pool were passed on to the next generation in one shape or form? Yes. But is it also true that 0.5% of the alleles just happened to, by random chance, not be involved in mating events and not be passed on to the next generation? Yes. This is called sampling error, and it gets worse and worse the smaller the population size or the smaller the number of coin flips. Just as you can have 7 heads and 3 tails with a very small number of coin flips, you can have very, very poor transmission of alleles due to nothing more than the small number of individuals in your population. This is sampling error. This type of genetic sampling error is what skews the allelic frequencies away from P and Q, away from the predicted and stable Hardy-Weinberg allelic frequencies we need in order to make the predictive math work. Here's a great example. Imagine you have a starting population. The population in this case is a bag of marbles. In this bag there are 50 white marbles and 50 green marbles. A relatively small population, an even smaller number of mating events. You choose 10 marbles out of that sack. Those marbles that you chose happen by random chance to be 60-40, or 6 gold marbles and 4 green ones. Slightly off from your true distribution of alleles in the population. But this is a mating event. So this becomes the next generation's allelic gene pool. What that means is we have to create a new sack of marbles that represents this allelic frequency. 60 white marbles, or gold, and 40 green. You then mate again for the second generation. You choose another 10 marbles out of there. This time you get 7 white and 3 green. But these are the marbles that mated into the next generation. So you reestablish a new sack of marbles that is 70-30. You draw from here. Now, by random happenstance, you got 4 white and 6 green. Those make it into the next generation. So we're kind of hovering all over the place, near 50-50, but not quite being 50-50 with each subsequent generation. Why? Because our number of marbles is relatively small, but even more importantly, the number of mating events is even smaller. So the alleles that are representative of that mating event are not quite entirely representative of all the alleles that were in the initial population. That's sampling error. Imagine a population where the allelic frequencies are 66% dominant and 34% recessive. But due to sampling error, 10% of the dominant alleles don't get passed on through matings. Well, 10% of 66 is 6. So the next generation is going to have allelic frequencies of 0.6 and 0.4. That's a marked difference, due only to a 10% skewing of mating events. We just changed allelic frequencies. The allelic frequencies in one generation was 66-34. The allelic frequencies in the next generation were 6 and 4. Hardy-Weinberg says that allelic frequencies cannot change. They do not change. They remain stable from one population to the next. They remain stable from one generation to the next. And obviously, with sampling error, that's not the case. We just saw allele frequencies change. Because our population wasn't big enough, and because our mating events weren't comprehensive enough, we experienced genetic sampling error. Genetically, this is called genetic drift. What's drifting is the allelic frequencies above and below the actual frequency. Because of, like marbles being pulled out of a bag, we are not quite getting the representative proportion of alleles in the next generation that mirror the current generation. Because this sampling error is random, you get the alleles you were dealt by those mating events, and that's all. It is impossible to predict. And the power of prediction is what makes Hardy-Weinberg so valuable. Genetic drift often looks like a population that's receiving migrants from some other population. It often looks like migration. It's just that there are no migrants shifting the allelic frequencies. The allelic frequencies are changing all by themselves because of the limited population size and the limited number of matings. All genetic drift is due to sampling error. That's the cause of genetic drift. But the reasons for this error can differ. Sometimes, populations can slowly and gradually reduce in their size due to limiting environments, such as a reduction of available food, water, shelter, etc. And then as these population sizes shrink, more and more dramatic sampling error occurs, more and more genetic drift is observed. There's also founder effect. Founder effect mirrors genetic drift. It's also due to sampling error, founder effect, but here there's true sampling going on. What happens for founder effect is a small subset, or sample, a small subset of an existing population sets off on its own and pioneers a new habitat, a new living niche, and sets up its own new population. In this case, we have about a 50-50 split of red ladybugs and yellow ladybugs. But only a collection, a subset of red ladybugs decided to set off on their own for greener pastures and set up a new colony somewhere else. This is a sampling error. We should have sampled the representative population and maintained the 50-50 split. But it was a random event that caused these ladybugs to just set off on their own and establish a new colony. And that random event just happened to, unpredictably, involve only red ladybugs. And so this entire new population of ladybugs are all red. They are the founders of this new population. But this allelic frequency in the new population doesn't mirror the original population because of sampling error. We didn't get a representative subset. We just happened to randomly get only red ladybugs. The allelic frequencies of this new population, regardless of how big it may grow on its own, will always be a representative allelic gene pool of the founders' alleles that they brought in, not of the entire original population. Only the founders established the new allelic frequencies because they were the ones that founded this new colony. The last example of how genetic drift can arise comes from a dramatic and sudden decrease in population size, not a gradual one. This is called a bottleneck. And bottlenecks give rise to very strong, almost population debilitating genetic drift. A bottleneck is when the number of individuals in a population radically and quickly, almost in a single generation, reduce in size very, very quickly. Northern elephant seals is a classic example of this. The number of northern elephant seals on this planet got down to about 20. About 20 individuals were left on this planet Earth. Had those 20 died, we would have had a full extinction. Through environmental programs, through legislation of many different countries, through a rallying of many different forces that was international, that was global rallying around these elephant seals, their numbers are back to be about 30,000. So the population is strong in number, but weak and unhealthy genetically, because the allelic frequencies, indeed the entire population of these 30,000 plus northern elephant seals, remains representative of whatever alleles happen to be in these 20. We were down to 20. The alleles in those 20 animals were all the alleles that were left on this planet for that species. And when this population rebounded to 30,000 strong, it did so only with the available alleles from this population. There are no other populations from which to tap into and get allelic diversity. Yes, mutations and evolution drives new alleles, but not at a rate that we can appreciate or see in so short a time. These northern elephant seals were poached. That's why their numbers got so low. And so we're talking about a pretty contemporary, pretty modern bottleneck for these animals. So genetic drift changes allele frequencies in a population, and it does so unpredictably. Hardy-Weinberg says that allelic frequencies must be stable, static, and predictable from one generation to another. So genetic drift invalidates Hardy-Weinberg. It's chance, pure, random, unpredictable chance, which alleles are going to make it through to the next generation when you have founder effect, when you have bottlenecks, even when you have population decline. In fact, from generation to generation, allelic frequencies increase and decrease randomly due to sampling error. They wander. They drift. Computer simulations can show this. In this computer simulation, there were 5 different populations that are explored computationally. Each of these populations started at 50-50 split of 2 alleles. 50% of the alleles were dominant, or A1, and 50% of the alleles were recessive, or A2. We have a 50-50 split of these 2 alleles. 5 different populations were started at this point and allowed to evolve from that point forward to see what genetic drift would cause. So this computational modeling of genetic drift among these 5 different populations, all starting at the same spot, showed how much these allelic frequencies can wander, how much they can drift from one point to another in the population. In fact, we can get allelic frequencies drift so much due to just sampling error, due to just the random mating, what alleles make it through to the next population and generation and which don't, that we get a allelic frequency reaching 1. Think of what that means. When an allelic frequency reaches 1, that means 100% of the population is made up of that one allele. Therefore, by definition, the allelic frequency of the other allele must become 0, because the sum of all the allelic frequencies is always 1. This is called fixation. In this case, for population 1, we see that the A2 allele has reached a frequency of 1. That means in this simulated population, there is no more A1 allele. It is gone. It has been bred out of the population. Same for here. Here, allele 2 has gone down to 0. Since allele 2's frequency has gone down to 0, the entire population is made up of allele 1. So here allele 2 became fixed, here allele 1 became fixed. And conversely, the other allele, whose frequency went down to 0, is said to be extinct. It's no longer available to the population. When an allele reaches fixation, and all the other alleles for that gene have become extinct, every single individual in that population is homozygotic for that one remaining allele. That means the population has suffered a loss of genetic diversity. There's no more heterozygotes because heterozygosity means 2 different alleles, and there ain't 2 different alleles. There's only 1. The only way to restore Other alleles is by mutation of that existing allele or migration of other individuals from a different population coming into this one. Fixation is actually a pretty common problem when there are small populations. Again, the smaller the population size, the larger the sampling error, the larger the genetic drift, and the more you drift, the more you run the risk of encountering fixation. All genetic drift leads to different populations of the same species exhibiting different allelic frequencies, eventually different phenotypes, and eventually speciation. If these populations continue to drift apart genetically, if their allelic frequencies continue to differ, they will eventually lose the ability to interbreed with one another. Now, these are two populations of the same species losing the ability to interbreed. Once that occurs, by definition, the two populations have become different species. The definition of a species is when individuals of the same species can mate and produce viable reproductive offspring. If two populations of the same species lose the ability to interbreed and produce viable reproductive offspring, they are no longer two populations of the same species. They are different species. Genetic drift, especially of small populations, is one of the driving forces behind speciation on this planet. We see an example of that in this salamander. Salamanders that were native to California, they started as a single ancestral species, a single ancestral population in Northern California. Slowly, over a great deal of time and many, many generations, they migrated southward towards the south part of the state. To do so, they encountered the mountain range there. The population split into two. Now again, two populations of the same species. They split into two at the peak of the mountain range, at the front of the mountain range, with some groups moving east and some groups moving west. It took a very long time, many, many generations, for these salamanders to completely migrate to the other side of that mountain range. By the time they had done so, and those two populations had met again, they were different species. They had lost the ability to intermate, because as those two populations were separate and they were small, they experienced a great deal of genetic drift. Their allelic frequencies changed across their entire genomes to such an extent that they could no longer interbreed with one another, and therefore, when meeting on the other side of the mountain range in California, these two salamander populations were now different species. Natural selection, evolution, also affects Hardy-Weinberg predictions in equilibrium. Natural selection is best described as the differential reproduction of genotypes. This is just another way of saying survival of the fittest, or reproduction of the fittest. This is obviously non-random mating. If more fit individuals are the ones that mate, we have non-random mating. We have positive assortative mating for fitness. And so, by definition, Darwinian evolution and natural selection breaks the rule of Hardy-Weinberg. We've discussed natural selection quite a bit, especially in the latter portion of the course, so I'm not really going to redefine it here. I'm going to trust that you're all pretty comfortable with the idea of natural selection and Darwinian evolution. But suffice it to say here that beneficial traits, traits that increase fitness and are genetically based, make it to the next generation more efficiently. The better and stronger mate more and pass their alleles to the next generation more. This skews allelic frequencies towards the better alleles. And the skewing of allelic frequencies from generation to generation flies in the face of Hardy-Weinberg. This is great for the species. This is the driving force of evolution. But it creates a problem for the math of Hardy-Weinberg. Believe it or not, mathematically, we can account for this. We can mathematically define fitness. We can put a number, a value, on fitness. This is the relative reproductive success of one genotype compared to another. And we can use that value, that mathematical value of fitness, to correct the Hardy-Weinberg equation. We can do that. That's done commonly in genetics, population genetics. We won't do it in this course because it's a little bit high level. We don't really need to delve that much into the math of it. The point is here that with natural selection, and therefore differential fitnesses between different genotypes and individuals, we can still mathematically predict the effects on allelic and genotypic frequencies as long as we have enough information about that fitness and we are correctly calculating for it in the math. We call this a general selection model, a mathematical model to predict allelic and genotypic frequencies accounting for fitness. And the results of selection vary, obviously, depending on what the natural selection is selecting for, depending on what genotype is the most fit. We'll go through a few examples. There's type 1 fitness, or type 1 in a general selection model. Type 1 selection means that the dominant allele is the allele that confers the fitness. So a homozygotic dominant individual is more fit than a recessive, but equally fit to the heterozygotic. Classic Mendelian dominance. If you're dominant, you're fit, and it doesn't matter why you're dominant, heterozygotic or homozygotic dominant. Because of this selection, the frequency of the dominant allele will gradually increase. We're selecting for it. The individuals with a dominant allele mate more, get their alleles to the next generation more, and so those individuals who are homozygotic recessive mate less, and the frequency of the recessive allele will decrease. This is called directional selection, selecting for a particular allele for directing the mating towards that allele. There's types 2, 3, and 4 selection. Types 2, 3, and 4 selection are also directional, but for type 2, there's selection against the dominant allele. For type 3, there is selection towards the intermediate heterozygote. And for type 4, there's selection against the intermediate heterozygote. This table's from your textbook, table 25.6, and it shows all of the 6 types we're about to discuss pretty succinctly and I think pretty clearly. So type 1. When you're type 1, being homozygotic for allele 1 or heterozygotic allele 1 is equal. As long as you have that dominant allele, you're good, and you're stronger and more fit than somebody who's homozygotic for the recessive allele 2. For type 2, we have the opposite scenario. It is the homozygotic recessive that is selected for, because there's increased fitness there, and that individual is more fit than a dominant individual. So whether you're heterozygotic dominant or homozygotic dominant, you are equal in your fitness but inferior to the homozygotic recessive. Types 3 and 4 rely on codominance or incomplete dominance. Here, the most fit is the homozygotic dominant. Less fit than the homozygotic dominant, but more fit than the homozygotic recessive is the heterozygote. So there's a hierarchy of fitness. The most fit individual is homozygotic dominant. Intermediate fitness is the heterozygote. Least fit is the homozygotic recessive. Type 4 selection is the opposite effect, where the most fit individual is homozygotic recessive. Slightly less fit than that is the heterozygote, but the least fit is homozygotic dominant. So in this case, there is incomplete dominance or codominance, making the heterozygote different in fitness than either of the homozygotic individuals. Excuse me one second, getting ahead of myself there. Eventually, for any of these cases, types 1 through 4, directional selection will lead to the extinction of the lesser alleles and the fixation of the best allele. So for type 1, we will eventually increase allele 1 to the point where it becomes fixed, and allele 2 is completely weeded out of the population and goes extinct. For type 2, it's allele 2 that will become fixed with the extinction of allele 1. For type 3, eventually allele 1 will become fixed, and for type 4, eventually allele 2 will become fixed. For types 5 and type 6, they are unique in that the heterozygote has its own individual fitness that differs completely from the fitness of the homozygotes. Type 5 selection is called overdominance. Here, the heterozygote is the most fit individual. So being heterozygotic makes you more fit than homozygotic dominant or homozygotic recessive. With overdominance, there is never allele extinction. Allele 1 and allele 2 will never become fixed in overdominance, and I want you to think about why. If you don't understand why, I want you to bring it up in your reflection. Try to figure out why there's never allele extinction or fixation with overdominance. Populations that are undergoing type 5 selection will eventually reach a Hardy-Weinberg equilibrium. Those allele frequencies will eventually balance out and become stable. Type 6 is called underdominance. In underdominance, the heterozygote has the lowest fitness value of all 3 genotypes. So being homozygotic dominant is more fit than being heterozygotic, and being homozygotic recessive is more fit than being heterozygotic. With underdominance, there is also never any allele extinction, and I want you to once again try to think about why. With underdominance, the population is actually constantly trying to rid itself of both alleles simultaneously. It's constantly trying to weed itself away from the heterozygotic condition. But because the two homozygotes are always being selected for, we have a hard time losing the heterozygotes. I want you to think about why. So let's come full circle and kind of wrap this lecture up by putting it in the perspective of our last lecture, when we discussed the power of Hardy-Weinberg math for predicting the behavior of populations. Hardy-Weinberg law came with a number of assumptions, a number of criteria that needed to be met. Very, very large size, random mating, no migration, no mutation, and no evolution. And our main question for today's lecture was why? Why is that so important for Hardy-Weinberg math? And the answer to that question is that the point of Hardy-Weinberg equations, the point of the Hardy-Weinberg law, is to allow allelic frequencies to be predicted generation from generation in a single population, and also, equally important, to allow genotypic frequencies to be calculated and predicted from those allelic frequencies. And small size, non-random mating, migration, mutation, and natural selection cause allele frequencies to be unstable and unpredictable. This figure from your textbook kind of summarizes much of what we talked about well, I think. The effects of mutation, migration, genetic drift, and natural selection, both in the short term. Of course, they all change allelic frequency in the short term. That's what makes them invalidate Hardy-Weinberg laws. But then the long-term effects differ, and this is what we talked about in the lecture. What are the long-term effects of each of these phenomena on population genetics? In some cases, these things cause allelic frequencies to be unstable in an unpredictable way. This is a good thing for life. Unpredictable allelic frequencies cause unpredictable fitnesses, and unpredictable fitnesses allow for natural selection to do its work, and overall can improve the health of a population. We need these disruptions to stability. We need these differences in our fitnesses. They balance themselves out. Within a single population, we have increased genetic variation due to mutation, migration, and some types of natural selection, some of the types we talked about in the previous slide. But we then counteract this and balance it with a decrease in genetic variation due to genetic drift and other types of natural selection. Between different populations, we can increase genetic variation with mutations, genetic drift, and some types of natural selection, but we can balance that by decreasing genetic variation between populations by such things as homogenization due to migration and other types of natural selection. So all of these disruptions to allelic frequencies, all of this unpredictability is a required ingredient for the success of a species long-term, evolutionarily speaking. But it is bad if you're trying to fit life into a math equation. What Hardy-Weinberg is trying to do is make those genetic processes that govern populations fit the Hardy-Weinberg equation. And that's beneficial for predictive value. But of course, all of these curveballs, all of these disruptions break the rules of that math. And so we, as geneticists, try to also balance the two and look at populations that come close to satisfying Hardy-Weinberg conditions, allowing for that genetic predictability, but also, of course, are subject to mutation, migration, and all of the other natural phenomena of life. So what did we talk about in this lecture? We started off just saying there are two broad categories of non-random mating in any species. Positive assortative mating, when individuals of a species mate with others that share the same trait or characteristic, and negative assortative mating, when different members of a population or a species mate with those that are different from themselves in a particular case. We used inbreeding as our example for non-random mating, for positive assortative mating for relatedness, simply because it affects all genes, not just one, and so the effects of non-random mating are amplified with inbreeding because it is a genome-wide effect. The increased appearance of lethal or deleterious traits that occur due to inbreeding are due to an increase in the number of homozygotic recessive individuals, and we refer to this as inbreeding depression. When species outcross, or mate with those that are not related to themselves, they counteract these effects and tend to be a much healthier population because they're getting much more genetic diversity and many more heterozygotes in that population. The influx of genes from one population into another is due to migration, when individuals from one population move to another and mate there. We also refer to this as gene flow. Gene flow or migration avoids speciation because it homogenizes two independent populations, kind of gets those alleles a little bit more comparable between those populations. It introduces new alleles and new allelic frequencies into what was once a previously stable population, but in doing so, it makes the allelic frequencies a little bit less predictable, and so they conform to Hardy-Weinberg a little bit worse. Once you have a population that's smaller than infinite, you have the potential for randomness skewing which alleles make it into the next generation. This is sampling error. We went through a number of different examples, both marbles in a bag as well as coin flips, to kind of isolate and explain sampling error. Sampling error in a genetic population leads to genetic drift, the changing or drifting of allelic frequencies occurring in that population. Genetic drift can occur for a number of reasons. One is just a gradual reduction in population size due to a gradual reduction in resources, but also founder effect and bottlenecks are special cases where genetic drift is especially pronounced because such a small number of individuals is establishing a new population. And then, of course, the allelic frequencies in that new population, regardless of how big it gets, is still a representative subset of the alleles that came in with those first founding individuals. When all other alleles are extinct, the one remaining allele that we are left with is fixed. We call that fixation. It has an allelic frequency of 1, and by definition, then every individual in that population is homozygotic for that one and only allele. That's what happens when genetic drift reaches its most dramatic conclusion. And then we moved on to natural selection. Natural selection is the differential reproduction of genotypes, right there refuting Hardy-Weinberg's assumption of non-random mating. Once we have a differential reproduction of genotypes, we have the skewing of allelic frequencies making it into the next generation. We skew the frequencies towards the better alleles that confer more fitness. There's a whole bunch of different ways that this can happen. Whenever particular alleles are selected for, we call that directional selection. You're increasing the frequency of those alleles from generation to generation, decreasing the frequency of the less fit alleles. And we went through 6 types of selection, 6 selection types to kind of emphasize this. And finally, we tied everything together and said that Hardy-Weinberg, its power is predicting allele frequencies in a population based on that population's stability and the stability of those allelic frequencies. Small population size, non-random mating, even natural selection, directional selection towards specific alleles, cause those allelic frequencies to be unstable, unpredictable, and therefore makes Hardy-Weinberg predictions impossible. And that's it. That is the end of the material for this course in genetics. I can't believe it. 19 lectures, 3 units, and a hell of a ride. So that's the end of the material for Exam 3. It's also the end of the material for the entire course. We will spend the rest of our time reviewing the 2 population genetics lectures in this upcoming week, and then we will move on to prepare for Exam 3 and, of course, prepare for the final exam as well. I always say thanks for watching, but I think this time it's appropriate to say thanks for watching all of these lectures, and I truly and genuinely hope you enjoyed this teaching and learning format.